How to access and explore data from ClinEpiDB.org, a guide for students. Are you a student of epidemiology, biostatistics, or global health looking for high-quality data for a project or to analyze for your thesis? We want to introduce you to our free, online, open-access data platform, ClinEpiDB. We integrate and facilitate access to datasets from global health research. For students and teachers of epidemiology, biostatistics, global health, and data science, we provide a source of rich, real-world datasets. These can be used as practice datasets to learn methods such as regression analysis, data modeling, and graphing. You can do secondary data analysis on these datasets for class projects or capstone projects, possibly resulting in publication. What is ClinEpiDB? ClinEpiDB is a free, online, open-access data resource. It's developed and maintained by scientists and software engineers based at several academic institutions around the world. Where does ClinEpiDB get data? We get data from global health researchers. Study teams partner with us to make their data available. There are several reasons why global health researchers share their data. It brings attention to their work, it benefits the field, and it is increasingly a requirement for getting research grants and for publishing their data. In this short presentation, we will demonstrate first how to find interesting datasets on ClinEpiDB, then how to explore data right in the browser, and finally how to access data for download so you can work with it on your own.
In this section, we will talk about ways to find datasets that are of interest to you. Start at the homepage ClinEpiDB.org. ClinEpiDB stands for Clinical Epidemiology Database. This is a resource where you can share, explore, and visualize clinical and epidemiological data. In the header, under Studies, you will be able to scroll through the list of datasets that are available on ClinEpiDB. We host over 30 datasets and release new data every couple of months, so check back frequently. Click on the Study Summaries table to get an overview of all of the datasets. It can take a few seconds for this table to load. As you scroll through this table, you can see that we host datasets that focus on different domains, including malaria and diarrheal disease. We host both observational and experimental datasets with a variety of study designs, including cohort studies, cross-sectional studies, and randomized controlled trials. By looking at the other columns, you can get an idea of the sample size of the study, the study site, and years during which the data was collected. This column, called Study Accessibility, tells you the level of access to the data that has been granted by the study team. Some datasets are public, meaning that everyone is free to download the data without restrictions. Some datasets are controlled. You have to sign in and fill out a data request form, and then you will be allowed to download the data immediately. Some datasets are protected, meaning that you will have to fill out a data request form which will be forwarded to the study team who will then approve your request within a few days. We will discuss data accessibility in greater detail in the last part of this video when we talk about downloading data. You can click this button to add or remove columns from this table. For instance, you could add additional data and WHO indicator subdomain. This adds 2 columns to the table. Let's talk about additional data. ClinEpiDB is part of a larger family of data resources called ViewPathDB. This includes genomic resources such as PlasmoDB for Plasmodium and VectorBase for arthropod vectors of disease such as mosquitoes. And MicrobiomeDB hosts microbiome data. So this column tells you whether participants from a particular dataset have data in one of our other resources. Here, the GEMS-1 case control study has additional data on MicrobiomeDB, and it is linked here. The WHO indicator subdomain tells you more about the focus of the study, such as whether it involves communicable disease or whether it's research on maternal and child health. So the study summaries table is a good way to identify a dataset that is interesting or relevant to you. And if you see something that you want to explore further, click on the study title to learn more about the dataset, or on this Explore and Analyze icon to look at the variables. Clicking on the title of the dataset will take you to a fresh analysis page where you can start exploring the data.
How to explore a dataset on ClinEpiDB. Once you find a dataset of interest, you will land on the dataset analysis page. We've mentioned before that ClinEpiDB is a data exploration platform, and this analysis page is the central location from where you can do a lot of things with this dataset. You can get an overview of the dataset. You can learn more about the context and methodology of this dataset. You can browse through all the variables and see the distribution of each. You can make plots. You can download the data. We are going to look at each of these things in some detail. But before doing that, let's consider, why would we want to explore data before downloading it? The first reason is that downloading data is a time-consuming task for you and potentially also for the original study team who may want to review data requests before granting you access. Before you spend time and effort, you should look at the variables, the participants included in the study, and the methods to see if this dataset is appropriate for your needs. A second reason is that our platform pulls together a lot of information from different sources, like codebooks, publications, and protocols. You can use this documentation to effectively learn about the dataset before diving into the data. A third reason is that our exploratory data analysis tools allow you to play with the data and come up with interesting questions to ask. You can query your hypothesis very simply in the browser, make some plots and tables, and see what trends may potentially exist before downloading the data and embarking on a more statistically rigorous analysis. And finally, examining the variables will help you decide whether you want to download the whole dataset or whether it will be more efficient for you to select a subset of data to download. With that said, let's explore the GEMS1 case control dataset as an example. At the top of each analysis page is a dataset diagram. This gives you an overview of the dataset. The first thing you can see is that the study team collected data on households and participants living in the households, and samples collected from the participants. You also see sample sizes. This was a large study with over 22,000 participants enrolled. And looking at the sample sizes, there appears to be one participant per household and one sample per participant. The presence of households and household repeated measures indicates that some data about the participant's household was collected only once, but other data was collected at more than one time point, which we group under repeated measures. Similarly, we have participants and participant repeated measures. Clearly, there is a longitudinal aspect to this study with data collected at multiple time points. So to summarize, the dataset diagram tells you the shape of the data and the sample size. What were they doing with these participants and samples? Let's go to the first tab here, View Study Details. This page holds a wealth of information about this study. At the top of the page, we link to a primary publication that you can read. As you scroll down, you will see a table of study characteristics. You can see at a glance that this study focused on diarrheal disease in children. Stool samples were collected. It was conducted in several countries, and the dataset contains nearly 600 variables. We can also see that this study has additional data in our sister site, MicrobiomeDB. Next, we have links to publications. We don't list all the publications from a study, but we select a few to get you started on a literature search. Then, if we have other datasets on ClinEpi that have data related to this project, we provide you links to those so you can check them out. Here we see that GEMS1 has a second follow-up case control study, and we also host the data from 2 community surveys. The next section gives you a summary of the background of this study. Why was it done? And the objectives of the study, listing the major research questions. For instance, the GEMS1 study was done to quantify the burden of diarrhea and to identify the microbial etiology of diarrhea. That is, to understand what pathogens are associated with diarrhea. And they wanted to look at these things across 3 age groups and across all of their study sites. Next, we see a brief methodology. This includes study sites, study design, and eligibility for cases and controls, telling you how they defined a diarrhea case and how controls were selected. There is a description of how data was collected. Here we see that at enrollment, a stool sample was collected from each case and control child. We also provide a lot of helpful study documentation that you can download, such as case report forms. The study team is listed on this page. Even when data is integrated on ClinEpiDB, the ownership of the data remains with the original study team. We merely facilitate access to the data. Now we have learned a little bit about the study and we are ready to look at the data. All the 596 variables in this dataset are available in the Browse and Subset tab. There are some variables that are important and highly likely to be useful in an analysis. We pull them out into a Featured Variables box. Let's click and look at a few of these. Country is an important variable in this study because it was conducted in 7 different countries. Country is a categorical variable. For categorical variables, we show you the univariate distribution with a frequency table and a bar chart. Here you can see that Bangladesh had 3,859 participants, corresponding to 17% of the participants from the study. The proportion of participants is fairly evenly distributed across sites, except Mozambique has fewer participants than other sites. Let's look at a couple of other categorical variables, like household study time point, which has 2 levels, enrollment and 60-day follow-up. The case or control participant variable is an important one in this study. Here we see that there are about 9,000 cases and about 13,000 controls. So the controls make up 58% of the total data set. We looked at a few categorical variables. What about a numeric variable like age, where the data are continuous? The univariate distribution of numeric variables is represented using a histogram. We provide summary statistics at the top. Here age is presented in months and the range goes from 0, that is birth, to 62 months, which is just about 5 years of age. And the mean age is 18 months. The featured variables box contains only a few variables, but all the other variables can be found in this variable tree underneath. You see that variables are grouped into categories which match the ones in the dataset diagram above. And the variables are organized into these categories. Under household, you can see variables related to geographical location, dwelling characteristics, dwelling facilities, household water, and so on. Open up any category to see the variables in it, and you can look at the distribution of each variable just as we did before. Let's look at the categorical variable water availability. Now this is the label that ClinEpiDB has assigned to this variable. The original names of this variable in the dataset are given here. We give it a simple human-readable name to make this variable easy to understand. We also provide the definition here that we pulled from the codebook. So this Browse and Subset tab is an interactive codebook. I should point out that there is a rigorous ontology underlying our data integration. What does this mean? In another study where similar data about water availability was collected, that variable will likely have a different name in the dataset. However, when we integrate data into ClinEpiDB, we give it the same harmonized label across datasets. So you can know that this variable is capturing the same data across different studies. Here, the frequency table and bar chart tells us that 68% of households in this study have water available all the time, and most of the rest have water available several hours a day. Up here, you can see that over 99% of households have data for this variable. So you can also get an idea of how much data is missing for each variable. When we have a group of related variables, sometimes we present them as a multi-filter. Let's look at an example. Drinking water source. This large group of variables asked about the main source of drinking water in the household. We combined it in this page, which lists the frequencies of over a dozen sources of drinking water. For example, you can see that 37% of households used a public tap as the main source of drinking water. Similarly, you can scroll down the variable tree and examine what variables were collected once on participants and what variables were collected at multiple time points on participants and what data we have on samples. You can search for variables too. For instance, you may wonder if the study team did any PCR testing on the samples in this study. You can search for PCR in the Find a Variable box, and it will restrict the list of variables to those containing the term PCR. So I can click here and see that they did PCR testing to detect viruses and bacteria in stool samples. A major question asked by the GEMS study was, what pathogens are associated with diarrhea? And we can see that many of these variables are found under the term stool microbiology test. They looked for different pathogens using a variety of tests, and the data tells us how many samples tested positive for a particular pathogen. In addition to looking at individual variables on this tab, you can filter data and examine a subset. For instance, in the country variable, you might want to look only at data from Asia. Select these 3 countries using the checkboxes. We now see that the counts in the dataset diagram automatically update to reflect the chosen subset, represented by the pink bars. A green filter symbol appears to indicate that the data has been filtered on a variable at the household level. A subsetted variable will appear as a chiclet, and hovering over it will display the chosen subset. Now that we have a filtered data set, if we look at another variable, for instance age group, we now see that the frequency table contains a column for the entire data set and also a column for our chosen subset. To choose a subset of numeric data such as age, simply type the desired range within the subset box. We can ask many interesting questions about this data. For instance, we want to explore the associations between these pathogens and other variables collected in the study. We can do this on the next tab, the Visualize tab. Click on New Visualization to open a menu of tools that you can select from. we offer the most commonly used visualization tools, including histogram, bar plot, scatter plot, line plot, and mosaic plot. Let's make a bar plot as an example and view the distribution of a categorical variable. This opens an empty plot, and I can choose the parameters. For a bar plot, the main axis has to be a categorical variable. And sure enough, something like age, which is a numeric or continuous variable, is grayed out and not allowed to be plotted here. Click on Expand All to see the full list of variables. You can move the slider to only show compatible variables, and then the list will be restricted to categorical variables in the case of the bar plot. I want to plot the prevalence of a pathogen, rotavirus. So click on the variable rotavirus by ELISA. The computation is done on the fly, and I get this custom plot. This shows that there were over 2,000 kids in this study that tested positive for rotavirus by the ELISA test. I can change the y-axis to show proportion instead of count. Now I see that 0.1 or 10% of the study participants were positive for rotavirus. You can stratify this plot by adding an overlay variable. Let's overlay this plot with the country of the participants. We can see some differences in rotavirus prevalence by country. Almost 20% of the participants from Mozambique are rotavirus positive. We might want to ask the question, is rotavirus infection associated with diarrhea? In other words, is the prevalence the same in cases or controls, or is it different? So we can change the overlay to case or control participant. The cases are shown here in dark pink and the controls are in blue. Over 18% of cases have rotavirus compared to less than 4% of controls. From this graph, it does look like rotavirus is associated with diarrhea in our initial exploration. We can further stratify this graph by adding a facet. Let's choose country as a facet. This splits our graph into 7 different graphs, one for each country. We can give this graph a name, and then if we minimize it, we see that it is saved in this space. So this was an example of how you can make graphs simply by pointing and clicking to explore associations between 2 or more variables. You can give your analysis a name and record notes about it. Your analysis is automatically saved in the workspace under My Analyses. If you are navigating ClinEpiDB as a guest, then the analysis will be lost when you close your browser. If you are logged into your ClinEpiDB account, then the analysis will always be saved in your account until you delete it. You will also have the option to share it with others. Making a ClinEpiDB account takes less than a minute. Use the link in the top right corner.
How to download data from ClinEpiDB. Let's talk about how to download data so you can work with it on your own. Datasets on ClinEpiDB have different levels of data access since the study teams who share their data always retain ownership and decide the level of access restrictions. On the Analysis page, access level is noted on the View Study Details tab and also on the Download tab. If you want to get an overview of data access levels, you can navigate to the Study Summaries table and look at the column called Data Accessibility. Let's sort by data accessibility and look at the different levels. Some datasets are public, meaning that anyone is free to download the data without restrictions, even as a guest. For example, the PRISM isomer cohort dataset has public data access. You will see a note on the Download tab of this dataset that says, "Data downloads for this study are public. You can download the data without logging in." Some datasets are controlled. You have to sign in to your ClinEpiDB account and fill out a data request form. This data request form may ask you a few questions, such as the purpose for which you are requesting data, your hypothesis, and your plans for data analysis and dissemination of results. Once you fill in and submit this form, you will be allowed to download the data immediately. The purpose field that you fill in will be publicly displayed in a table on the View Study Details tab to help the study team keep track of requests and to promote collaboration between researchers with similar interests. Some datasets are protected with more stringent requirements. For these datasets, you will have to fill out and submit a data request form. This will be forwarded to the study team to review and approve, and our team will notify you when you are granted download access. In some cases, we may write back to you and ask for more information about your data request. This approval process generally takes 3 to 5 business days, but it may take longer at times. You can always write to us at help@clinepidb.org to check on the status of your request. Once you have access, our Download tab allows you to download either a customized subset of data or the entire dataset. Let's start by looking at how to download a subset of data. You can customize the observations or rows in the data by filtering via the Browse and Subset tab. For instance, if you select data from only the 3 countries in Asia, you see that in the Download tab, the number of households, participants, samples, and repeated measures are reduced to only include data from the countries you selected. If you want to download participant-level data, click on this link to open up a modal. The first step is to select the variables that you want to download. Key identifiers, such as participant and household ID, are required. You can then look through the variable tree to select all or a desired subset of variables. As you select variables, you see them appear in the table. Once you are satisfied with that table, you can click on this button to download the data. It will be saved as a text file, and you can open it in programs such as Excel, SPSS, SAS, or R. If you want to download the entire dataset, you can use the links under the full dataset. The README files give you useful information and tips for working with the data. Use key identifiers such as household and participant ID to merge files as needed for your analysis. This completes our tour of the ClinEpiDB workspace. For more help with ClinEpiDB, please write to us at help@clinepidb.org. You can also find helpful tutorials under the Help drop-down menu in the header. To keep up with our webinars, announcements, and releases, follow us on Twitter at ClinEpiDB, or just keep an eye on the right sidebar of our homepage. Thanks for watching this video.